Hello, welcome to another bioinformatics video. In this video, I'll be going over the exercises related to BLAST and multiple sequence alignment. So in the first problem, we are given some unknown fragments of a sample that supposedly comes from a pathogen, and we're told that the fragments are all derived from one protein, but that some of these sequences are actually random, and we should try to find out the protein of interest in the sample by using BLAST. So I'll just run at the beginning of this exercise. We should install BioPython. And to run this local version of BLAST, we'll be using a database, SwissProt, which we must download and then unpack on the Google server. And we're importing some homemade libraries that are partly based on the book by Rocha and Ferreira. And yes, and we have a Blossom table. And that is everything we need. So what we're going to do is simply make a loop. And the idea with this loop is that we'll go through each fragment in the list of sequences we have, and it will run a BLAST, run this mini BLAST on it. And this mini BLAST, it will not perform the final step, which is to do a full pairwise local alignment via the Smith-Waterman algorithm. And so it's up to us to do that as well. And we'll do this alignment for all of the BLAST results that are produced. Now, what is a BLAST result? So we have we have a database which we've imported here through this function read database FASTA. So we've just applied the file path to our SwissProt database that we just uncompressed, and it will be stored in a dictionary called DB. And to look up a record in this database, we just simply need to supply a number. So the records are numbered. And the result of this BLAST best alignment FASTA algorithm here, the result of this will be a list of numbers of records in the database. So using this result string here, we can go into the database with a particular number in mind and fetch the record that's stored there. And among other things inside the record is, in fact, the sequence. And from the sequence, we can get a pairwise local alignment by looking at the database entries sequence aligned with the fragment that we're interested in. And of course, supply to it a Blossom matrix that we'll need for doing the alignment. And at the end, we'll format the alignment so that we can read them well. And this will use this pairwise2.format alignment that we used in a previous exercise, which takes as its inputs the 5 particular outputs of the pairwise2.align.local ds function. So the overall structure, just to review, is we have this big loop, the outer loop, which is going through each fragment and producing BLAST results. And then we have an inner loop, which is going through each result, since multiple results are produced, and fetching the individual records from the database and displaying them after doing Smith-Waterman local alignment. And the idea is, so I won't run this yet because it takes a couple seconds. The idea is that the aligned sequences will share a common protein. So if we take a look here, let's see, can we scroll here? Yeah. So if we take a look here, and it takes a bit of exploration of these sequences, and if we just kind of scan through them, we'll see a few good hits. So here is a good hit, and we can recognize it immediately because it's mostly matches here in this aligned region. If we take a look at the other alignments, we can see that they're random, that they're just, yeah, they're not good hits. And oops. And we see the name here of this particular good hit, envelope glycoprotein GP160. If we keep scrolling through, here's another one. So here we can see a hit, which happens to have a lot of matches. It has the same name. We can see envelope protein, glycoprotein GP160. Let's continue scrolling. And we're seeing a lot of random hits still. But the idea is that we should see good hits. And every now and then we can see also by the score. So here this score is 513. Even though it's not in the view here, we have probably a good feeling about this one, that if we scroll down, and yes, and sure enough, here we get an alignment with quite a number of decent matches. We can continue. And again, just checking the scores. Here's a score that's 696. It's also to this envelope glycoprotein GP160. If we scroll down, we see a lot of hits. And it's interesting also that we're hitting different parts of the sequence. So we're starting to get a sense that this really is our fragments, that they're fragments of this protein, and we're getting, depending on which one we look at, different matches and different positions of the envelope glycoprotein 160. So this is, in fact, the answer. This is our protein. And for the curious, we can also scroll through some of the other information in this record. And we see this tag here, HIV1. In fact, envelope glycoprotein GB160 is the spike protein, basically, of the HIV virus. That mediates the attachment and entry of these virus particles into human cells. So that is the solution to our puzzle. That's the identity of the pathogen, and that's the protein of interest. And now I'll just run this, and we can see that it does, in fact, take a while for everything to run. And the first hits will start to trickle in. So we'll move on to the next problem in the meantime. So problem 3.2 simply asked us to replace the sequences that were already present in the examples that had been given. So what was that exactly? So if we look, so we're given this list of Cas9 related sequences. So these sequences, they come from this original study from 2014, and they come from different sources, different bacteria, but they're all similarly displaying some conserved regions. So the idea is we'll use multiple sequence alignment to look at some of the similarities between these sequences that come from very different origins, but nonetheless share a common function. And so we have this large block here, and the idea behind this block is simply to perform the multiple sequence alignment. And the idea behind this was not really so much to think so hard about the actual code going on here, The idea behind this problem was more to focus for a change on the biology and on the output rather than making a programming puzzle out of it. But I can explain some of it anyway. So we have the sequences here as a dictionary. And for this particular function, we would like to supply it just the sequences without their IDs here. So there is a method associated with dictionary objects in Python, which is called values. And we can call this method like so, and this will just return a list of all the values in the dictionary. Likewise, there's a corresponding one for keys. So if we typed cas9 seeks.keys, we would get a list of all of these guys here. So that's what's happening here. And then this particular multiple sequence alignment module, which is a bit partially homemade, it has its own sequence object, quite similar to the BioPython sequence object, but not exactly the same. And to run this particular multiple alignment function, we should convert our sequences up here into this particular sequence object. So this this structure here is something called a list comprehension, and this is basically just a way to to construct a list using a for loop. And so this is a bit of an advanced structure, so you don't really need to know this, those of you in this course. But what it will do is it will create a sequence for each value in the values list, and it will return this list then of the sequences that are this particular protein sequence object from this multiple sequence alignment module. But anyway, long-winded explanation, but basically this is just a list of our sequences, and we can then feed this to the multiple alignment function down here. The other stuff up here is also just some preliminary stuff. So we're importing the blossom matrix, which is saved in these two lines here. And here we're initializing the parameters for pairwise alignment, which gets used during the process of the multiple sequence alignment. Basically, it's running multiple pairwise alignments at a time. So that's all this line does. And by the way, we can check on our blast to see how it's going. So it's still running, but we can see some of the results have printed out now. But that is taking a while. And then, so yeah, so this will produce the multiple sequence alignment. And at the end, we have, I'm just going to stop this one up here so that we can move on. So we have at the end this multiple sequence alignment object. And what is this exactly? So if we just type this in, oops, I have to run this. And yes, so excuse me, and we have this multiple sequence alignment object. And in fact, all the data is stored in here. So all of the information about our gapped multiple sequence alignment is contained in this object, but it won't just print itself out if we just run this. So we instead need another few lines here, which will be devoted to accessing the data that's in this object, and that's what this block is for. And basically, all it's going to do is go through each sequence in the object, and the object has attached to it a method called align consensus, which will just pull out the gapped sequences in a list form. And for each sequence, it will prepare a sequence record, which can be used to be saved as a FASTA file. So BioPython has a module called seek.io, IO as an input-output, and this module is just devoted to reading and writing files. So the idea behind this block is to read all the sequences, create record objects, and then save them as a FASTA file. And we briefly mentioned somewhere, where is it? Up here, yeah, so the FASTA format is simply like this. It's the sequence down here, and then at the top, a greater than sign, and the name or identifying information. So that's all it's going to do, basically. It's all that identifying information is contained up here. So we got the name and the sequence. Here's an ID, a name, a description. These can all be saved in the FASTA format in that top line by the greater than sign. But bio.seek.io will handle all that for us. So all we have to do is prepare the record and give it a file name. So that's what this block does. And while we're doing it, we print out our sequences. And so that's what we have down here. And if we print out the sequences from this alignment, which have gaps inserted, if we print them all out in order like this, line by line, this actually becomes our multiple sequence alignment. And you can even start to see conserved columns here of conserved amino acids. So each position, each position here corresponds to a position in the multiple sequence alignment. And this last panel down here is all about visualizing the same information. So no new information is really added here. This additional step, running another function, view alignment, is utilizing some graphical libraries in Python to create this interactive plot. And if you click it, you can drag it and scroll through, as you can see. And we've color-coded the base types, the residue types, according to amino acid, to better visualize it. And we can see there's also like a kind of global map of all the sequences up here from 0 to the end. And just looking at it, you can see conserved regions. So, here is a kind of conserved region. Here's a kind of conserved region. And the problem just asks us, what is the base at position 99? So here's position 100. Position 99 is this one right next to it. And we can see it's a couple S's, a gap, but mostly G's. So the most represented base here is a G. So that's it for this problem. So the main step was really just copying and pasting in the sequences here, and then everything else was simply a matter of running the respective blocks here. And finally, problem 3.3, which we had some problems with in the initial writing of this problem. So I apologize for the mistakes, but I have updated it now. So I will go through the way this problem was meant to be completed with everything where it's supposed to be. So we have this set of sequences that come from this NSP3E nucleic acid binding domain of beta coronaviruses. And this family includes, of course, SARS-CoV-2, but it also contains sequences from other coronavirus members. And this is often the idea with these multiple sequence alignments, is we have a family of related but not identical sequences that we're interested in. We're trying to understand some of the conserved substructures in the main in the protein. So we have this big list here. And we have this handy function, which we'll use later, to count the number of amino acids in a list. But then we have this big list here, and this is running just like the other multiple sequence alignment cells that we showed up here, except that at the end, this one is producing a list called the sequences, and the sequences will simply be a list of strings, and each string is our gapped sequence for that particular number for that particular original sequence. And the error originally in the problem was here. So we had accidentally called one of the earlier list of sequences. So to do this problem properly, we should add the correct pointer here to these sequences. So we'll run this, and we get this large multiple sequence alignment. We can see some conserved columns here. And the problem in this case is to count the amino acid sequences in a particular column. So it asks us to find, let's see, it asks us to find the consensus sequence and include any gaps. So we want to count the amino acids in each column and then print out a sequence that shows the winner basically for each position. And we're going to use this little function at the top here that counts amino acid sequences. So the real trick in this problem then is to figure out a way to slice this data in a nice way, because we're given basically a list of strings. So if we check the sequences and we put a 0 here, this is going to be one string. And it's not a numpy array, so there's not an option to just say like, I want, all rows column 1. That would be nice. If we had a numpy array, we could fetch a column. So numpy lets us do that, but instead it's just a list of strings. So there's no way we can fetch a column here. Python just understands these as separate string objects and not really connected in any way. So this is the way we can get around this, is to essentially convert it into a numpy array. So we start out with an empty list here, and we're going to go through each of the sequences. So we have a for loop here for seek in the sequences, and then we're going to add a listified version of that string to our empty list here called separated. So if we have a string, whatever, and we run the list function, it will convert the string into a list with each character being separated into elements in the list. So that this is, by the way, just one way that you can do this problem. So there are probably multiple approaches, but this is one way to go about it. So then at the end of these few lines, we'll have this list called separated here, and it's split all the. See, we have a huge list here. It's split all the amino acids up into elements. Okay, so, and I'll just delete this here because it takes a lot of space. And then what we're going to do is convert that list of lists, right? Because each one of these is a list, and we put it in an additional set of brackets here to make that a list of a list. And then we're appending that to the sequence here. So the output of this thing is really a list of lists. And each of those lists has individual bases in them. But then what we can do is take this list of lists as the input to a numpy array function here. And if you remember, when we supply a list of lists to np.array, numpy.array, this will convert it into a matrix, basically a multi-dimensional numpy array. So then as soon as we've done that, created this matrix, all of a sudden our data becomes much easier to navigate. So if we print this out, now we have this, and it's being truncated here because it's huge. So we have the dots here, but we have a matrix. And we can slice through it if we want to fetch, say, the first row, all columns. We could do that. So here is one row. And if we go up to the top, we can see indeed that it is this row here, right? T-I-K-Y-S, T-I-K-Y-S. Let's say we wanted to fetch instead of column, numpy is good for that. We can fetch column 0 in much the same way. So now we got TPPT. If we take a look here, going down, we see TPPT. And then this type of data is then all we need to feed to our little frequency counting function here, which takes a list or numpy array and simply spits out a number, which is the most prevalent base. So let's do that then. So we've got this here. Here we're going to make an empty consensus string that will grow as we go through each column. And then we have a for loop here, a range loop that is going to go through every column position, basically. So numbers from 0 up to the number of columns. How do you get the number of columns? Well, it's simply the length of one row, right? So we have row 0, all columns. You could also easily put the length of one of the strings, one of these original sequences here. That would work as well. They should be the same. And then the column values then are going to follow the same form that we use here, except instead of a 0, we're going to replace 0 with our column iterator here so that it's allowed to change. So on each iteration through, it's going to be fetching all rows of some column, basically fetching the column in the form of a list, in the form of an array. And then we simply need to feed it to our little function, most frequent, have this list of values, and then we get our most prevalent base as the output of that. And then we can just tack this onto our consensus. So we add this plus equals, and we'll then increment the growing consensus here that way. So here we have the consensus being printed out. And it looks like this: I, K, A, Q, F, K, and so on. So that's it for that problem. Finally, the optional challenge problem: generate an amino acid frequency table like those discussed in the lecture. Use the multiple alignment data from the zinc finger example earlier in this section. Your matrix should have a row for every amino acid type and a column corresponding to each column in the multiple alignment. And elements of the matrix should have values corresponding to the amino acid frequency in that column. So I just went ahead and copied back the zinc finger sequence set, and we'll just run this exactly like we did the first time with the zinc finger sequences. At the end, I'm putting everything into a variable called zinc seeks. And this is like the last problem, going to call this align consensus method from the multiple alignment object and just assign this set of sequences to a list here. So if we print them out, we get going to run this. Maybe like this, list. Yeah, there we go. So yeah, we can just print them out and we can iterate through them as well. So in this case, we want to not just get the most prevalent base as we did in the consensus problem. In this case, we want to fetch the frequency of every base that is presented here. And so since we're dealing with every base or every amino acid, the first thing we're going to do is get a list of all the possible amino acids. And then we're going to want to go through each sequence and do much the same thing that we did in the last one. So fetch a column by transforming all this data into a numpy array. So the first half of this, the first 2/3 of this actually looks exactly the same as our solution to problem 3 here. So we're just going to take the data. We do the same thing with the separated sequences, converting them first into a list of lists, feeding that list of lists to a numpy array to create a matrix, and creating, yeah, and then using the matrix properties of indexing that lets us fetch, say, all rows and one column. We use that then to fetch all the values in a column. So we'll have a similar for loop that goes through each position, each column, and then this column values thing, which is going to pull those amino acids from the column. So just like the last problem, except this time we're going to create a matrix called freqmat or frequency matrix, and it's initialized as a zeros matrix. So it's going to have the dimensions equal to the number of amino acids and the length of the sequence. So we'll have one position for every position in the multiple sequence alignment and one row for every type of amino acid. So this is our frequency matrix. And then we're going to go through each of these columns and run this function here, get freaks, and add as input the column values that we extracted here. So kind of similar to running this get most frequent function that we had for the third problem, except this one is going to work a bit differently. So the way it's set up is we have an amino acid list here and a counts list initialized as zeros for each of the amino acids in the same length. And then it's going to get zipped into a dictionary. And then we'll simply go through each base in the column of amino acids and then look them up in the dictionary here. So basically finding the appropriate key, which is going to be some amino acid, and then incrementing whatever value is stored there by 1. So this can be a way then to count the amino acids in a particular column. And then after doing that for all column values, it will then return in list form the values of this dictionary. So coming out of this get freaks function call down here is just going to be a list of values, which are the counts basically of each amino acid type. And then we simply want to take this data then and update our freqmat matrix up here in the appropriate column. So we use the index here to get to the right column and the AA or amino acid index to get to the correct amino acid. And then we're simply going to store there whatever value is next in this list. So using the amino acid index in that case. And then finally, we can just print out a visualization of the matrix or print it out down here. So here we can see our frequency table. Oops. And we have to declare this variable, of course. And down here we have the printed out frequency matrix. So yes, and we can check. Yeah, I believe the first value here is for gaps. So we have a lot of gaps. That tends to be quite frequent. So that's what we're looking at in this first row here, I believe. Yeah. And then the second amino acid is A. So I think if we were to check, we should find 1A in the 0, 1, 2, the column 2. Yes, I believe that is this A here. So yeah, indeed, we're fetching the counts here. And if we wanted to, we could convert these frequencies into fractions by dividing by the number of sequences. That would be fine. Okay, and that's all for this video then. So I'll see you in the next video.